So let me switch gears. So thank you, Mike. I mean, it was a great talk. Really appreciate it. So my name is Raghu Machiraju. I'm a professor of computer science and engineering and pathology and bioinformatics. I work very closely with Anil. And so my job right now is to introduce, a very good friend whom I've known for many years. Okay, Anant Madabhushi. So I'm going to introduce him, and I'm sure it's going to be a great talk. So, um, do Carly, is that you in charge? Do you have the slides, or should I bring up my own slides? Okay, I think I'll just bring up my own slides. Okay, I wasn't sure. Okay. Okay, Amir is doing. Oh, great, Amir. Okay, thank you. So, um, Anand has been very active in this space ever since he became a professor. Actually, we almost got him at Ohio State, but he decided to go elsewhere. So I know Anand since those days. So he's right now a Donald Institute Professor of Biomedical Engineering at Case Western. So he's also, sort of reading from the slide, but I'll be a little selective. And he's also the director of the university's Center for Computational Imaging and Personalized Diagnostics. I mean, he has many appointments throughout the university, and he certainly has very strong connections with Cleveland Clinic and also with the Veterans Affairs establishment. Now, let me say a little bit more about his work. So it's all about making machines smart so that they can identify certain diseases. He's focused a lot on lung cancer, and he's been very well recognized. So, in fact, Nature magazine called him out as one of the five scientists who have applied truly offbeat and innovative approaches towards cancer research. So he's been singled out, right, for his work on using AI to towards health disparities, to addressing health disparities. And he has many I guess accomplishments to his credit, manuscripts, patients, patents, and so on and so forth. I'm sure you can read all of that on the web pages. But it's so in closing, I think it's really a pleasure to introduce Anand, and we look forward to hearing your talk, Anand.
Thank you so much, Raghu. Very generous, overtly flattering introduction there. Thank you very much. Just have to warn all the folks. Unfortunately, it's going to be all downhill from here on. So I'm just putting that out there. Anyway, so I just wanted to talk today a little bit around Pathomics as a potential alternative to deep learning to prognosticating disease outcome. And apart from my collaboration or affiliation with Case Western Reserve University, just a quick shout out to the Department of VA. Since 2019, I'm also a 5/8 research scientist, federal employee at the VA. So also working on developing and applying AI approaches for the health and betterment of our veterans. Like my good friend and preceding presenter Mike Wiesett said, no conflict, no interest. So I'm also happy to report that we are working with a number of industry collaborators to help move some of the developments and discoveries we're making in the lab, out towards translation and towards deployment. If I'm completely honest, I guess I would confess that my true disclosure is the fact that I'm also on social media, and very unashamedly, I am here broadcasting my social media Twitter handle. If any of you are on Twitter, you like any of what you see here today, please feel free to give me a follow. So I want to set up the problem a little bit by first providing some statistics that I'm sure a lot of you are familiar with. The lifetime probability of a man in the United States being diagnosed with some form of cancer is about 50%. For women, it's only a little better. About 1 in 3 women will be diagnosed with some form of cancer in her lifetime, meaning that about 40% of the US population will be diagnosed with some form of cancer. Now, when you look at the actual mortality on account of cancer, there is a disconnect between the diagnostic incidence of cancer and the number of people who are dying because of the various types of cancers on an annual basis. Now, this is not to suggest that 600,000 deaths on account of cancer is a small number by any stretch of the imagination, but it does bring home the point that there is a disconnect between that diagnostic incidence rate and the mortality. Now, we know that certain factors have helped reduce that mortality rate. We are more aggressive with screening, more aggressive use of imaging for early detection, better use of biomarkers. But we have to be also cognizant of the fact that certainly one of the reasons why the mortality statistics are discordant with respect to the diagnostic statistics is because we are overdiagnosing and overtreating a large number of cancers today. And prostate cancer is a classic case in point where 1 in 6, 1 in 7 men will be diagnosed with prostate cancer in his lifetime in the US, but only 1 in 40 will actually die on account of it. And we know that prostate cancer is a classic case in point where we overtreat and overdiagnose a number of men, subjecting them to radical therapies like prostatectomy or radiation therapy, when in most cases these men are likely to have indolent disease. And not only does the overtreatment cause real harm to our patients, there is also the other unintended consequence of financial toxicity that overdiagnosis and overtreatment causes. And the statistics that I'm sharing here today, I'm sure a lot of you have seen, but I mean, it's quite mind-boggling when you think of the fact that 42% of new cancer patients will lose their life savings. And certainly, like I said, a certain responsibility to the statistic is the fact that we are overdiagnosing and overtreating so many cancers. So what I want to talk about today is the role of digital pathomics and computational pathology, not just from a diagnostic standpoint, so not just in terms of better decision support tools for the pathologist, but really how can we develop better AI tools to help in prognosticating disease outcome, predicting progression of disease, predicting response to treatment. In other words, how can we think of AI, how can we think of computational pathology as a companion diagnostic for the clinician? How can we think of these tools being used by the oncologist or the clinician to really tailor the therapy for a given patient based on their specific risk profile? And how can AI play a role in helping to realize this promise of precision medicine? Of course, I'm sure pretty much everybody in the audience is well aware of Pathomics, the term that's become popular to describe computational feature analysis and extraction of features from digitized pathology images, the ability to use machine learning and computational image analysis tools to go in and identify individual cell types, individual primitives from these pathology images, to look at a series of different subvisual descriptors that capture the spatial architecture of the primitives, to look at the shape, the appearance, the textural patterns, the heterogeneity patterns within different tissue compartments, within the stroma, within the epithelium. And the great thing about Pathomics is that you can convert this whole slide image into a digital, non-tissue destructive biomarker that now provides far more granular information about the biology of the disease, about potentially the progression of the disease, as well as treatment response, compared to just visual assessment of these whole slide images. And so about a dozen years ago, myself and Sridhar Ganesan, Mike Frellman, John Tomaszewski sort of had this idea that if we could leverage these tools, we could start to pull out patterns and features from H&E images that could then potentially serve as a non-destructive, a tissue non-destructive companion diagnostic. So one of the questions we were looking at that time was trying to look at early stage ER positive breast cancers and try to figure out whether women with early stage disease could successfully avoid adjuvant chemotherapy, because we knew that in 70% of these women, adjuvant chemotherapy would really not have any added benefit. Now, there are molecular tests to try to address this issue, but these are expensive, they're tissue destructive, and they don't always capture the riskiest portion of the tumor. And so our idea was that essentially by developing this IBRIS tool, you had a clinically non-destructive way of interrogating the tumor, prizing out patterns and features from the whole slide images, and then being able to connect those patterns to disease outcome and to treatment response. and essentially therefore have a much larger impact than molecular diagnostic tools that are much more expensive. So that was sort of the fundamental idea. So how do you go about constructing these tools? Well, you've heard quite a bit about deep learning and neural networks, and I'm not going to belabor the point, but just as just one example from some of our work from about 6 years ago, where we stood up a neural network, particularly a stack sparse autoencoder for detecting nuclei from pathology images. As I'm sure all of you are aware, you have multiple different architectures, multiple different configurations of these neural networks, but fundamentally the idea behind most of them is that you're providing a deeply annotated set of examples to the network. The network, in an unsupervised way, is learning representations that best separate out the target of interest from everything else. And even though it may not be clear what exactly these representations are, the huge benefit of these approaches is the fact that with minimal training, with minimal knowledge of the domain, you can stand up these predictors and be able to identify the targets of interest. So in this particular paper, we were able to demonstrate that we could we could provide annotated examples of nuclei in whole slide images to the neural network. The neural network was then able to go in and segment out the nuclei in a pretty quick, efficient fashion. A few years ago, we then decided to challenge the neural networks and said, well, could we challenge neural networks to do more than just segmentation? So this was back in 2018, where working with a group at Penn, we developed a deep learning algorithm to identify risk of heart failure from whole slide images of H&E tissue biopsies. And so we did it in the classical machine learning mode. We had a set of training images where the machine classifier knew what the categories of interest were, whether these were patients with heart failure or not. We had a separate holdout test set of patients where the classifier was not exposed to those patients. And when we ran the algorithm to predict risk of heart failure from the endomyocardial biopsies on the holdout test set, we found that the algorithm had an area under the curve of 0.97. So we were really thrilled by this. And what was also interesting was that the comparative performance for two pathologists who were looking at the same test set was only about 0.74. So there was a 23% differential. We got all very excited, put out a press release. That got some traction. I was briefly trending on Reddit. So it was all hunky-dory. But the postscript to that story is that a couple of months later, when we got the next tranche of images from Penn. Now remember, the same lab, same scanner, same demographics. The next tranche of images, when we ran it through the classifier tanked, right? So the classifier went from 97% AUC down to 75%. And we couldn't figure out, for the life of me, I couldn't figure out what was going on. Thankfully, I have really smart students I have the honor and privilege of working with. And so they were able to figure out that what had happened between the first tranche of images and second tranche was that a remote software upgrade had been applied to the slide scanner. And that had very imperceptibly changed the appearance of the whole slide images. And because of that, the neural network had difficulty with the second tranche of images compared to the first tranche. And so this really brought home a really critical point that we have to be very aware of the fact that when you've got these representations that are abstract and you don't understand, because of the opacity with these representations, when you don't understand how and where and why the algorithms are converging on their predictions, then it becomes very difficult to go back and try to ameliorate the problem, to try to troubleshoot and figure out what went wrong. And this is a theme that is getting a lot of traction. A number of other people are talking about this. This is a quote from Professor Jordan at UC Berkeley, a real giant in machine learning, who's mentioned that trusting these brute force algorithms too much is a fate misplaced. Cynthia Rudin at Duke University had a very influential paper she wrote in Nature Machine Intelligence a couple of years ago, where she said that we just have to stop trusting the black box, particularly for high stakes decision. And this is an example I've shared many, many times in my talks, but it's a very useful one to really reiterate and reinforce the issues with the black box approach. And it's an example of work from Samir Singh's group at UC Irvine, where he and a student trained a neural network to distinguish huskies from wolves. And what they found was that for this problem, the network had a 99% accuracy. And so Samir, who clearly is way smarter than I am, was extremely skeptical of these results because he realized that the neural network couldn't be doing that well because huskies and wolves looked very, very similar to each other. What they were able to then figure out was that the network wasn't actually learning any features of the face of the animal. It was simply learning the fact that huskies invariably were on a white background. There was snow in the background. And that's what the network was picking up. It wasn't actually picking up new features of the face of the animal. It was that background, which in itself is not an issue, but it's an issue when you don't understand or appreciate what the representations mean or where they're coming from. So I just want to make that point. But at the same time, I don't want to just trash talk deep learning, right? I mean, there's a huge advantage of these approaches. And the way we have looked at these approaches in our group has really been from the perspective of segmentation and identification of primitives. Some work that we put out where we've shown that these approaches such as generative adversarial networks can be very valuable in going and segmenting out individual cells, lymphocytes, macrophages. So not only can you do segmentation, you can also do classification pretty well and really do deep phenotyping of the cellular microenvironment of tumors. And beyond that, also by compartmentalizing and identifying the different tissue compartments, stroma from the epithelium, you can also add geographical tags, right, for the individual cells. So you can not only identify a lymphocyte or a tumor-infiltrating lymphocyte by compartmentalizing and partitioning of the different tissue, different tissues in the slide. You can also figure out whether it's a stromal tail or an intraepithelial tail. So one is able to then assign, like I said, these geographical tags. But then beyond that, one can start to look at trying to characterize in a quantitative way the spatial arrangement and the spatial architecture of these different cell types within the different tissue compartments. And so the fundamental premise here, I mean, the fundamental takeaway from this talk, I hope, will be the fact that by focusing on these features that are a little bit more interpretable, that are related back to the pathobiology of the domain, you have an opportunity to prognosticate outcome, predict therapeutic response, and do it in a way that is up there with the deep learning approaches, but with the added advantage that these are more intuitive, more interpretable, but critically are connecting back to the pathobiology of the disease. So one example of one of these handcrafted features that we've been working on that we're very excited about is looking at the polarity of collagen fibers. So this is in the area of breast cancer, where we've developed approaches to go in and segment out individual collagen fibers. Haojia Lee, who's a graduate student in my group, has been able to assign a vector that looks at the orientation of the collagen fibers with respect to the tumor nest. So what you see in green is the tumor nest that she's been able to identify with machine learning. And then by borrowing an idea from high school physics, namely the concept of entropy, we're able to assign a number that captures the relative order or disorder of the collagen fiber orientation in these tissue slide images. So what you see here is that in patients who tend to have short-term survival, the collagen fibers are all very well oriented with respect to each other, and therefore the entropy is low. On the other hand, when you look at patients who have long-term survival, that is, they do well, the degree of disorder is much higher. The entropy is much higher. The collagen fiber orientation is more discordant with respect to the edge of the tumor nest. And so this seems a little counterintuitive. You would imagine that patients who tend to do poorly, the collagen fiber is going to be more disordered. But it turns out that collagen potentially provides the vehicle or the highway for cancer cells to hitch a ride and progress from the primary site to other metastatic sites. On the other hand, if you look at patients who tend to do well, the haphazard nature and the chaotic arrangement of the collagen fibers impedes the progression of the cancer cells, cancer nuclei, from the site of the primary to other sites. And therefore, almost the disorder in the collagen fiber orientation, therefore, is potentially a good biomarker, a favorable prognostic biomarker for outcome. So in work that's currently under review, we've demonstrated now in over 400 patients, both from the TCGA as well as ECOG 2197, which is a ECOG actor clinical trial conducted back in the mid-90s, for ER-positive breast cancer, that we could prognosticate risk of recurrence in these women just based off the polarity or the disorder of the collagen fiber orientation. And just to sort of quickly recap here, we're just talking about standard H&E images, no fancy imaging, no fancy microscopy, just digital slide images of the H&Es, and the rest of it is these computationally derived image features that relate to the polarity or the disorder in the polarity of the collagen fibers that we're demonstrating as prognostic. In more recent work, we've demonstrated that even other features that more directly connect to what pathologists tend to use on a daily basis are also extremely useful. We've known for the longest time that Bloom-Richardson grading or the Nottingham grading scheme is very, very powerful, particularly for ER-positive breast cancers. We know that the grade of the disease is strongly associated with risk of recurrence and prognosis. The challenge with grading, and in fact, in most grading schemes, is the fact that you don't have very good reproducibility or agreement between different pathologies. And so Yuli Chen, a visiting scientist in our group, took up the challenge and developed an algorithm to go in and quantify the different hallmarks of the Bloom-Richardson grading scheme by using computational analysis approaches. So essentially, she was able to go in and find a set of descriptors, mathematical descriptors, that captured nuclear morphism, so looking at variance in the shape, size, arrangement of the individual nuclei. She was able to come up with an index that captured quantitatively the number of tubules on the whole slide images, and similarly was also able to create a mitotic index. And when she took all these different parameters and combined them, lo and behold, we found that not only in lymph node negative ER-positive breast cancers, but also in lymph node positive ER-positive breast cancers, we were able to prognosticate risk of recurrence across a number of patients from a number of different sites. And so this is from university hospitals, this is the ECOG clinical trial data set that I mentioned, but also working with our partners in India, the Tata Cancer Center Memorial Hospital in Mumbai, we were able to demonstrate that we were able to prognosticate risk of recurrence in patients from Tata. So not only did it work for patients in Cleveland or patients who had undergone clinical trials here in the US, but it was working also in patients from another continent. And so this was really, really gratifying. But it's not just prognosticating recurrence or risk of recurrence in invasive disease. One of the other areas we've been looking at is DCIS, or ductal carcinoma in situ, where features relating to nuclear shape, disorder of the orientation of the nuclei, and the texture appearance of the nuclei is also strongly prognostic of risk of progression from DCIS to invasive ductal carcinoma. And this is a really critical question because, as I'm sure a lot of you are aware, the big challenge, the big clinical conundrum in DCIS is knowing which of these patients should receive adjuvant radiation therapy and which patients can be spared adjuvant radiation therapy. And so what we've done is been able to use a completely computational pathology-based approach to risk stratify which patients will progress or not, and therefore potentially identify patients who should be the beneficiary of adjuvant radiation therapy versus those patients who could successfully avoid adjuvant radiation therapy. Another area that we've been spending a lot of time on is in the area of prostate cancer, specifically predicting risk of recurrence in prostate cancer. Similar to DCIS, one of the big questions in prostate cancer is after surgery, we know that about 20% of men will have recurrence. And we know that a number of men who have biochemical recurrence or disease recurrence after surgery are at high risk for metastasis and therefore are at higher risk for prostate cancer specific mortality. So the big question is knowing after surgery, which of these patients should benefit or should be getting adjuvant therapy, either docetaxel or radiation therapy. And so Patrick Leo, a graduate student in my group, has now demonstrated in almost 1,000 patients from 6 different sites that features relating to gland shape, gland morphology, gland appearance is prognostic of disease recurrence in all comers, but also critically, when you look at margin negative patients, or patients who've got surgical margins that is bereft of cancer, even in those patients that typically tend to be lower risk, even in those patients, these approaches are strongly prognostic. You can see a hazard ratio of 3.01, suggesting that we can actually identify those patients, even though they're margin negative and lower risk, which patients potentially might be candidates for either adjuvant docetaxel or adjuvant radiation therapy. Now, there is a molecular diagnostic test called the Decipher, which is a 22 gene expression based assay in the NCCN guidelines for predicting risk of metastasis in prostate cancer patients after surgery. But it's a $3,000 test. It's a tissue destructive test. And we did a recent study where in 167 patients, we went head to head between the image score, the image classifier, and the gene expression based test, and found that there was non-inferiority. That is, a completely non-destructive test, tissue non-destructive test that really costs pennies on the dollars compared to this $3,000 test was non-inferior in terms of prognosticating risk of recurrence after surgery. But what we also did was we took two very simple clinical parameters, PSA, prostate-specific antigen, as well as the Gleason score. When we took those two things and combined it with the image features, that is, we created a nomogram, we found that we had a hazard ratio that almost doubled what was possible with the Decipher test. So even though head-to-head, the image classifier was non-inferior to Decipher, when we took the image classifier and combined it with these two clinical variables, we were now able to do significantly better in prognosticating risk of recurrence compared to the Decipher test. And so this is something we're very, very excited about, because now you've got a non-tissue destructive test with two parameters that seems to be outperforming, not just non-inferior, but outperforming a $3,000 test that's in the NCCN guidelines. But it's not just nuclear morphology or gland morphology. One of the things we've also been really interested in is looking at lymphocytes and looking at the spatial arrangement and spatial architecture of tumor-infiltrating lymphocytes. In a paper we published a couple of years ago, we demonstrated that it wasn't just the density of tumor-infiltrating lymphocytes, but the spatial arrangement, the co-arrangement of tumor-infiltrating lymphocytes, the ones you see in green here, identified by the machine algorithms, with respect to the cancer cells in red. That interplay was quite different between early stage lung cancer patients who did have recurrence from those patients, patients who did not have recurrence from the patients who did have recurrence. So the actual spatial co-arrangement was quite different between these two categories of tumors, and that's reflected in these Kaplan-Meier curves. In more recent work, we've delved further deeper to try to understand what's going on with these TILs. And so working with Kurt Schalper and David Rim at Yale, we did a study with looking at adenocarcinomas as well as squamous cell carcinomas and found two interesting things. One was that the actual spatial TIL patterns that were prognostic were actually different between adenocarcinomas and squamous cell cancers. Adenocarcinomas tended to be characterized by a density, a larger density of TILs that was more prognostic. In squamous cell cancers, it was more the spatial co-arrangement of cancer cells and TILs. But what we also did was had access to quantitative immunofluorescence images. So we were able to do subtyping of the TILs and did a co-registration of the H&E and the QIF images. And when we did that co-registration on a cell-by-cell basis, we were able to map whether an individual TIL was CD4 positive or CD8 positive or CD20 positive, and found that even there, from a molecular standpoint, adenocarcinomas tended to be characterized by CD4 positive cells, but in squamous cell cancers, it was CD4 positivity and CD8 positivity that tended to dominate. So my point is here is that not only is it important to find these prognostic features, but to the extent possible, the molecular interpretability is also important to really provide as much transparency as we can to these prognostic predictive signatures. In abstract we presented at ASCO last year, we demonstrated that the spatial TIL motif was also predictive of response to immunotherapy in gynecologic cancers, and that this, looking at endometrial, cervical, and ovarian cancers, we were able to prognosticate overall survival based on the spatial TIL motif in a cohort of 50 cancers. More recently, working with Jim Lewis's group at Vanderbilt, we've demonstrated in almost 1,000 patients that the spatial TIL motif is also prognostic of outcome in P16 positive oropharyngeal squamous cell carcinomas. Critically, I just want to point out that one of the things that we did very deliberately do in this study, the study took a long time to pull together, not just because we needed a lot of patients, but one of the things that we really wanted to do was to demonstrate that this signature was robust across multiple sites. So we worked really hard.
Can I just remind you that it's already.
I'm going to wrap up. Just one more minute.
Yeah. Take your time. Yeah. Okay. Go ahead, please.
And so the critical thing that we wanted to do was to demonstrate the resilience and robustness of the signature across 6 different sites. And then finally, we also combined the spatial TIL signature with multinucleation, which is aggregates of these large nuclei, and demonstrated that in combination within individual AJCC 8th edition staging groups, that this combination was prognostic of disease-free survival. One last thing I'll leave you with is some of the work we published last year, where we demonstrated that when you looked at the morphological features of nuclei within the stroma between different populations, so specifically we looked at African Americans and Caucasian Americans with prostate cancer, there were significant differences in the appearance of the nuclear morphology between the two populations. And we used that to build population-specific prognostic models for prostate cancer, and found that a population-specific model was much more prognostic of recurrence in African Americans compared to a model that didn't explicitly account for those differences. So meaning that when you think about these AI approaches, we have to be thinking of these other factors. We might need to think about gender, we might need to think about population-specific differences, ethnicity-specific differences, and avoid or resist the temptation to sort of just throw the kitchen sink at these approaches. So with that, I just want to end. So hopefully what I've conveyed is that there's a real opportunity for using computational analytics with routinely acquired H&E images to address questions in precision medicine. Hopefully what I've conveyed is that the low-cost aspect of these tools can really have global impact, especially in low and middle-income countries. And then finally, hopefully what I've conveyed is that there is really a need to also establish the intuitive basis of these approaches to drive clinical adoption. And my 2 cents is that's going to be an important driver. The connection between these image features and morphology and molecular features is going to be an important driver of the use of these tools in the clinic. Just want to thank the amazing students, scientists, faculty, colleagues, and postdocs that I have the pleasure and honor to work with and are responsible for the data I showed today. And of course, we always thank the people who put food on the table, thanking funding agencies, the DoD, as well as the NCI and the NIH for their generous funding. Thank you very much.
Thank you very much, Anant. That was a great talk. So I think we're really out of time, but maybe one question, a micro question if somebody has, and if there are other questions, maybe you could put it on the chat and Anant will respond. Anybody? So I think Anil has a question. Okay, how can the average pathologist in Akron use those tools, questions for pathologists and computer scientists? So maybe we'll just limit it to pathologists, Anil. Okay, because we're out of, well, computational pathologists like Anant. So how do you respond to that, Anant?
Yeah, so I think that I think I'm going to build off what Michael said in his conclusion, which is that one is really making the pathology community more aware. Of course, I think the folks on this particular call are all drinking the Kool-Aid. I don't think they need to be convinced, but just explaining and making the other pathologists aware of these kinds of approaches. Now, one of the things I didn't get to a chance to talk about today, but we're very actively working on, are tools to help democratize the process of machine learning, right? So very often, one of the challenges that we've identified has been that there is this real bottleneck, right? You need, the pathologist wants to use these tools. Maybe the pathologist has seen some of this and is eager to engage, but they don't know how to. So how do you go about doing this? You're going to work with a data scientist, you're going to work with a computer scientist, bioinformatician. Maybe they don't exist. They're a fairly rare commodity still. But if you can start to develop approaches that allow a pathologist sitting at her or his workstation to start to stand up a neural network very quickly with a few annotations, a few examples. In other words, if you provide them the fishing rod to go and fish, then I think you can start to create these tools literally sitting at your cubicle, at your workstation and stand these things up. And I think we're getting there. I think we're getting to the point where we can start to create the infrastructure to help democratize the process of standing up these neural network strategies so that you almost sever the bond or the dependency to some extent of the pathologist's reliance on the data scientist, on the computer science. And this is not to say that you want to do away with the collaboration. Collaboration is critical, right? But I'm talking about things where you want to find, you want to train a model to do lymph node mets or something, or mitotic figures or something like that. And you don't have, maybe at the University of Akron, you don't have somebody working in computational pathology. So that's sort of the opportunity where I think that these tools, which will really help folks who might not otherwise have been able to use these tools to now really get into it. They don't need to know they don't need to know Python. Don't need to do coding, but they understand how to interact with these tools and stand up these networks. And I think that's going to be, in my mind, going forward, is probably going to be a important factor that's going to help further disseminate and increase awareness and interest and engagement. And I think this is the point that Michael made, is engagement. Get more pathologists engaged in the process. And I think these kinds of tools will help facilitate that.
Thank you, Anant. And